All right, we're going to be talking about the beta-amyloid story. And I'll start out, first of all, talking about the role of Etheresia Pretorius, PhD lady from South Africa, Douglas Kell, PhD guy from England. They did a lot of great work on the concept of amyloid microclots, or fibrinolytic clots they're called. They're also called microclots, 2 to 200 microns that plug up your small vasculature. They're associated with poor dentition, leaky gingiva, and leaky gut, whereby the bacterial endotoxins, LPS, lipopolysaccharide from gram-negative bacteria, LTA, lipoteichoic acid from gram-positive bacteria, get into your blood, distort the shape of fibrinogen, have it undergo an amyloid transition, and then it forms a lot of small clots. Okay, let me just show you what a little bit better sense of what that looks like. Oh, first of all, I'll talk to you briefly just about scabies. So scabies is a disease, let's say in sheep, where the animal has intense itching, so it just scratches itself and it scratches off its coat there. You start out with a normal configuration of a protein, which is designated, protein, and then C next to it for cellular. Once it changed to the prion form, the SC stands for scabies. That's a more energetically stable position. It's not going to go back to its original normal folded configuration. Okay. Now here is what you're talking about with an amyloid transformation. This is going to come up a lot. So once you know it, it's not that big of a deal. Every protein is formed and then it folds into its natural shape. So fibrinogen, blood clotting protein, should be cylindrical, like it's called alpha helix secondary protein structure. Primary structure is just a sequence of amino acids. Secondary protein structure is sort of a choice between alpha helix, beta pleated sheath, for example. And then tertiary structure is the overall shape of it. Quaternary is when it's shared with another one. And like tertiary would also be, distant combinations like disulfide bonds and that sort of thing. Anyways, so to change from being like a slinky, cylindrical in shape, to being flat like beta pleated sheets is a big deal, because you can't stack cylinders very effectively like slinkies. They don't stack well in biology. But once everything is flat, they become very sticky, and you can keep on sticking more and more of these things together, beta pleated sheets of individual protein monomers, and you combine them by hydrogen bonding, and they stack like a deck of cards. And once they form a big enough molecule, the higher the molecular weight in aqueous solution, like the plasma of the blood, the more likely it's going to precipitate. To precipitate is to clot. So the bacterial endotoxins will do it. Also being iron overloaded will do it, like from eating red meat, from eating processed foods that are fortified with iron. Person becomes iron overloaded, men usually in their 20s, women in their 50s. All of these things predispose to increased clotting, and increased clotting is a big deal. That's why people die, get sick and die. In the movies, people die, from gunshot wound when they bleed out on the carpet. Okay, there's the drama. Okay, but in real life, people die from clotting. All right. Typical old person, they all have the same disease: hypertension, diabetes. They plug up their arteries, causing atherosclerosis. They plug up their arteries of their brain. They become stupid. To their heart, they get AFib, congestive heart failure, heart attacks. So anyways, leaky gut causes this because these bacterial endotoxins get in the blood. Being iron overload causes it. Viral infections can cause this as well. Okay, so that is amyloid clotting, and also called fibrinolytic clotting. You need to know about that. If you want to understand human health, that's an important thing to know, and 99% of conventional medical trained doctors don't know this. Okay, some of the areas it starts getting a little interesting. Now, here's another article we just saw this group here, Douglas Kell and Etheresia Pretorius. Iron dysregulation and dormant microbes as causative agents for impaired blood rheology, okay, making the blood thicker. So when you get increased free iron in the blood, bacteria need that to grow. You also get reactivation of dormant bacteria, and those can cause problems in the blood, and they can also release their endotoxins, okay? And then systemic inflammation and oxidative stress will increase blood-brain barrier permeability. You don't want that, because then that permits bad things to get into your brain. All right, the next guy we're going to talk about is this guy, Jack Audie. He has done a lot of good research on beta-amyloid, okay? And he's done a lot of research on neuroinflammation and some important neuroinflammatory chemicals like NF-kappa B and IL-1B. I'll explain what this all means in just a moment. NLR, P3 inflammasome, NOD-like receptor, pyrene 3 inflammasome. Okay, I'll explain what it all means, but he's a really good teacher. He's got YouTube videos too. He's a very bright guy. He's done some great research. Okay, first thing to know about is how the amyloid precursor protein is cut. So this is APP for amyloid precursor protein. It's a cell membrane, transmembrane, spans the entire membrane protein. Okay, under normal conditions, it's cleaved by what is called alpha secretase. It'll cut it in half like that, and that's what you want to have happen. That does not lead to any amyloid problems. Okay, on the other hand, if it gets cut by B for the bad secretase, that'll cut just above it, and then there's a gamma secretase cuts just below it. Then that will release the amyloid peptide. So just the individual little, it's called beta-amyloid 42. I think it's about less than 50 amino acids consecutive is called a peptide. Over 50 it would be called a protein. So any of these individual peptides by themselves, not a big deal, but they'll start to form oligomers. Okay, so just several of them attached to each other. Over time, they can eventually form polymers, much more of them attached to each other, insoluble fibrils. Okay, and these will cause a whole bunch of different assorted problems. But this is where it comes from, amyloid precursor protein. Aluminum will cause more of the bad type of cleavage. Just to give you a sense of some of the bad things that amyloid precursor protein can do, it will cause beta amyloid protein to bind to the NMDA receptor, which is a glutamate receptor on the postsynaptic neuron, and it'll increase the opening of it, so more calcium will get into the cell. Because more calcium gets into the cell, you'll have more activation of this postsynaptic neuron. What that means is it's an excitotoxin. Normally glutamate opens up this channel, the NMDA receptor, but glyphosate can do it, the stuff sprayed on non-organic soy, aspartame, that artificial sweetener, also called Nutrici, and beta amyloid protein will open up. Magnesium deficiency will also increase. All of those things will increase opening and activation of this NMDA receptor, allowing more calcium into the cell. The more calcium goes into the cytoplasm, the more the postsynaptic neuron gets activated. The important point to know here is that beta amyloid protein is toxic. It overactivates the neuron that it binds to, and then what that means is more calcium gets in the cytoplasm, so more ATP is going to have to be taken from the mitochondria and used to run the pumps to get that calcium out of the cytoplasm so that cell can turn itself off and get back to rest. That's a real important point. And the more you overstimulate a cell, the more chance you run of exhausting its energy production capacity, its ATP, and that's going to have real serious consequences and kill the cell potentially. Okay, one thing you need to know about is what's called phosphatidylserine externalization. So phosphatidylserine here is the phospholipid with the red color to code it. Okay, normally the phosphatidylserines are supposed to be kept in the inner leaflet of the plasma membrane of a cell. The more they go to the outer leaflet, the more likely that cell is going to die soon. It stiffens the plasma membrane of a red blood cell, so it can't pass through the spleen, narrow diameter capillaries, and it gets phagocytized, chewed up by the spleen macrophages. But in brain cells, accumulating phosphatidylserine in the outer leaflet is like an eat me poster, a neon sign telling the brain microglia, brain macrophages, to eat it. So we're going to come to that in just a moment, how that all works. And there's a reason for it. Okay, so the enzymes working with the phosphatidylserine system are flipase and flopase. So these are well-named enzymes. And phosphatidylserine on the outer leaflet is an eat me signal. Okay, here is a microglia. Excuse me, these are the guard cells of the brain. When they're at rest, they're ramified, meaning they have branches. Rama means like branch. Okay? As they get primed, that means partially activated. They start becoming more round. And when they're fully activated, they're like round. They look like a scrambled egg appearance. And once these guys start releasing their toxic chemicals, they can release very toxic chemicals, including hydrogen peroxide, excessive amounts of nitric oxide. And we're not talking about the small amounts, released in an artery for vasodilation. We're talking about big amounts that can actually be quite toxic in that context. Okay, plasma phosphatidylserine normally on inner leaflet, phosphatidylserine on the outer leaflets and eat me signal. Okay, let me show you a picture of it now. So here is the phosphatidylserine with the red, the phospholipid head. And normally the enzyme flippase keeps it flipped correctly on the inner leaflet of the plasma membrane. Okay. You also want to flip phosphatidylcholine to the outer leaflet. And it takes ATP to do that. Both of these enzymes require ATP, and the cell does this on purpose because it's an indicator of whether or not the cell has enough ATP. Okay, if the cell does not have enough energy, it can't maintain phosphatidylserine to be flipped to the inner leaflet of the plasma membrane, and that's a problem. Excuse me. Phosphatidylserine, then when it gets flipped into the outer leaflet, that's a big neon sign saying, eat me. So the microglia, the macrophages of the brain, the guard cells of the brain, they will come and they will eat that neuron. The reason why it's done by this phosphatidylserine flipping into the outer leaflet is so that it exactly indicates that cell. Because neurons, there's millions of them all crowded close together. If it was just a chemical secreted into the extracellular space, the microglia would not know exactly what neuron it was coming from. But because it's the actual plasma membrane of the cell that's run out of ATP, the microglia knows what to eat, what to ingest. But you can see this is bad. That neuron could have an important memory stored on it, and now it's getting eaten. You don't want that to happen. Well, what's going to deplete that ATP? Something that overstimulates the neuron and wastes its ATP for nothing. Something that inhibits its mitochondria so it can't make that much ATP. Here's just showing a few other things that contribute to this problem. Excess stimulants, using up the energy supply, SERCA inhibitors decreasing the ability to pump calcium out of cytoplasm, mitochondria inhibitors decreasing the ability to make ATP, atherosclerosis making the cell hypoxic so it can't make so much ATP. If the person is hypotensive, they don't get enough blood to their brain, hypoglycemic, don't get enough glucose to their brain, hypoxic, don't get enough oxygen to their brain. All of those things will decrease the energy production ability of that cell. So this can happen. Okay. All right, I think we'll take a break here. So that'll be the end of part 1. I hope you found that helpful. We're probably going to do about 3 or 4 parts. There's still a lot of information to cover.